So I think we're going to get started here momentarily if you want to find a seat. Right?
Shri is supposed to be kind of like announcing people, so I'll let him know.
Okay. Next ones. I'll introduce Frank. Do you want to put my slides on? Well, no, they're there, but they're not up there.
So they're going to, in the back, the audiovisual guys are going to take care of switching it over.
Okay, so if you want to put my slides up.
Okay, perfect.
Excellent. So welcome. It's great to see a lot of familiar faces. For those I haven't met, my name is John Day, and I'm a neuromuscular neurologist here at Stanford and have the good fortune of working in both our pediatric and adult clinics. This is the first time we've done something quite like this. We have had a number of disease-specific conferences, and we'll continue to have those. But I think there's also a lot that can be learned between ourselves by doing something broader. And so we had this idea of trying to bring a larger group together. I think there's wisdom amongst you all living with different forms of neuromuscular disability or problems in movement. And I think that there's a lot of information that can be shared. So we're really excited by the turnout. We're excited to have you here. We're eager to do this on an annual basis going forward, in addition to continuing to have more focused conferences when we have specific issues to attend to. But I really would appreciate getting any kind of feedback that we can. So please don't be shy about telling us things that you wish we had covered, you really want us to cover in future years. This isn't a one and done kind of a thing. The idea here is that we'll try to set this up to be a recurring conference that we can have on an annual basis and hopefully learn from each other in a continuing way. Furthermore, that allows us to try to capture the energy that we get by just a critical mass of people involved. The more people that we can show are engaged and involved, that allows us to interact with the university and the hospitals in ways, gives us some leverage with the pharmaceutical companies and organizations to work with them to make sure that we're doing more to help everybody with these conditions. So it's really great to see you all. And it's really exciting for me to introduce the people in this program. We set this up, I don't remember when we set it up, Jennifer, 6 months ago, more than that. We've been talking about it and chose a date and got everything set. And now I've got to leave. So the oddity is that this field is so incredibly active now that there's just a lot going on, and unfortunately I have to be in Argentina tomorrow afternoon. So I'm glad I can be here to launch this, but I apologize for butting out early, and want you to know that you're in phenomenal hands. So we have a number of people. I can see a lot of people at this table that are part of our team, but there are a lot of people part of the team. I guess one thing we can do in future years is wear a sash or something so that you know who's who. And I really encourage you to corner anybody, kind of buttonhole them and ask them questions or tell them things that you have always wanted to tell people, because we're all here to learn. None of us thinks we know it all. We're all eager to learn from each other. And really, again, appreciate your coming here with us. So I just thought I'd give you a little bit of an overview of the concept here. Is our concept, as we've been building this program, is to take really a comprehensive approach to conquering neuromuscular disorders. And what does that mean? It means that we obviously need to have superlative clinical care, and we've worked hard to accomplish that and are continuing to work to refine and improve that and strengthen that. We need to couple that clinical care with research and training. So I'm quite proud of the neuromuscular fellows that we're getting. We're getting some of the best people from across the country now to participate with us, and that moves our whole program forward. And then we're going to work with the community to make sure that we're doing everything we can. We think it's our responsibility not just to provide medical care, not just to undertake research, but to work with the community and make sure that we're advocating and accomplishing what's really going to be meaningful by working with you all. So we started with very good clinical care, and Stanford is world-renowned for its bench research, but the two weren't always interacting much. And then we had some clinical research, but not very much. That's grown, so that all three of those have grown. One impacts the other. And then we've increased our teaching and outreach, which really act as a conduit for all of them. As we see more patients in clinic, as we work hard to take care of more patients, that gives us access to information so that we can take blood or cells or specimens from patients and give those to the Stanford research community so that they can get more engaged in helping us work out things, that comes up with new ideas that we can trial in clinical trials. And because we have a group of patients now that we're working with, we can actually carry out those trials. And these things have continued to grow, and consequently the strength of our program has continued to grow, and we're not done yet. We're continuing to expand and to strengthen our program. We're not going to take over the entire university, but we're going to work hard to make sure that we're capturing the interests of some of the brightest and most productive biomedical researchers in the world who are on campus, so that we're really working to connect the dots, and that's kind of the concept. But none of this happens without people. And so we're excited that we have accumulated really a world-class group of people that are working together to accomplish this. So this is our neuromuscular team, and many of whom you will meet with today in various ways. And again, I encourage you to avail yourself of their presence and talk to them, or complain about things, or do whatever, to help us do more for you. So we're very excited about this. Today's program is going to start out with Frank Bennett from Ionis Pharmaceuticals. It used to be ISIS Pharmaceuticals, and for some reason they changed their name. So they moved from an Egyptian goddess to a Greek god, and all has gone swimmingly since. And really, it is a tremendous honor to have Frank here. Frank, as far as I'm concerned, is at the absolute pinnacle of biomedical research development for treating neuromuscular disease. He spearheaded the work. Some of you will have heard of the drug, some of you are on the drug, called Spinraza, and he's been working on that for more than 10 years as part of his work on that whole type of drug, which has gone on for several decades, so that he really brings a phenomenal expertise to the field and has garnered all kinds of awards for his work. And it's really exciting to have him here, because this is an important point. that even though a treatment is necessarily developed for one disease, the principle of that treatment validates is appropriate for almost all genetic disorders. So that's very true of Spinraza. So that it indicates that it works, and now it's a matter of adapting it so that we can use it for any number of other disorders. So it's very exciting. When a research trial doesn't work, there are about 10,000 reasons why it might not work. When a research trial works, as Spinraza did phenomenally well, that means all 10,000 of those things had to have worked. We learn a tremendous amount when we can see something work as well as that worked. And with that understanding, with that foundation, now we can move on, and are moving on as rapidly as possible to use that approach to treat other neuromuscular disorders. And so all of those things are going on, and it'll be exciting to hear Frank's talk. That'll be followed by one of our own. Sri Muppidi is going to introduce you to the inflammatory and immunological aspects of disease. You might not realize it. You've probably heard of polymyositis or inflammatory neuropathies or myasthenia gravis, which are inflammatory and immune-mediated diseases, but all disease has an immunologic aspect to it. So muscular dystrophy, there's inflammation of the muscle. In spinal muscular atrophy, there's inflammation probably in the spinal cord. In ALS, there's inflammation in the spinal cord or brain. So that inflammation is a part of all these conditions, and it's really critical that we learn how to control it so that we can maximally impact these. And then Tina Duong will be talking about living with these conditions and how to optimize your health. One of the things that is absolutely true is that we need to maintain our health, maintain our functioning as well as possible while we're waiting for these new treatments, but even after that. I mean, you know, it's not a magic wand. You know, these conditions don't just disappear. So it's a matter of learning how to live optimally with that. And Tina brings a phenomenal expertise in exercise, and we'll be able to talk about that. And then this afternoon, we're going to break into smaller groups so that you can talk one-on-one with some of our team members, but also with each other, so that I think we can begin this networking process so you can learn from each other and we can learn from you, so that Jacinda Sampson will be talking with myotonic dystrophy and myotonic disorders, patients with those. Sri will be talking with patients with inflammatory conditions. Dr. Yuen So, an internationally recognized ALS expert, will be talking about ALS and other motor neuron disorders. Carly Siskind, who's widely known, internationally recognized for her work on CMT, will be helping with that. Dr. Jenna Klotz, one of our prized new members of our faculty, will be talking about Duchenne and primary muscle disorders. And then we have others coming in. So Lorna Drope talking about important issues with trusts and conservatorships. Again, this is what we're thinking, that there are common elements that are going to be important across these conditions that we want to explore. Sarah Strandberg, who I worked with for many years in Minnesota, is here to talk about swallowing, a common issue in neuromuscular disease. And then we have some others here from the Independent Living Group to talk about aspects of community care and support, and then on getting and using durable medical equipment. So we tried to set this up so that it works well, but again, please give us feedback for the upcoming years. So what can you do to help us fight? Well, being here is a huge part, so I thank you for that, and hope that we can maintain this relationship and continue to expand it. You can register if you haven't before with our database at the Stanford table out front. We have some of your names, but we'd love to have all of you registered so that if issues come up pertinent specifically to you, we can contact you. That'll be in our database. It'll get you access to some of our trials. We have a lot of trials going on in many different conditions, and they can talk with you about that at the table. And then donations, and by donations, obviously money's always good, but I'm also talking about donations of biopsy specimens or surgical specimens. If you completely end up, not needing your body anymore, we'd take that as well. But seriously, I mean, there are things we can only discover, unfortunately, by autopsies, and we have a whole program for that. We actually have one of the premier sites for that in the world that we use this tissue to support research around the world on these conditions. And then feedback. So I've said it about 16 times. We'd love to hear from you what works, what doesn't work, and what you want us to do in the future. So thanks again for coming. It's great to have you here, and without further ado, I'm eager to get Frank Bennett up on stage so we can hear the exciting things that are going on at Ionis Pharmaceuticals. So thank you.
It's okay.
You want to, that one?
That one.
Or 2 clicks? 1 click, 2 clicks?
We'll see.
Oh, there we go.
Yeah.
Great, thanks
Good.
again, Frank.
Sure.
Really appreciate it.
Should be just like a few minutes or so.
I'm sorry.
They're going to bring it up. It should be a few minutes.
Oh. So first off, I'd like to thank John for inviting me up here. It's really a pleasure to present some of the work that we're doing to this audience. It's I'm presenting to scientists, and I have a habit of getting into the weeds. So if I start using terminology in my talk that you're not familiar with, please raise your hand. I'll try to not use too many scientific jargons in what we're in my talk. But first off, I really do appreciate you guys being here. It's important for us as a company that's developing drugs to better understand what patients are dealing with in their diseases. And so your presence today is really important for both us as well as the clinicians and caretakers at Stanford. So just as John mentioned, I'm an employee of Ionis Pharmaceuticals. And so, I'm an evil drug company, I guess, is one way to think about it. But really what my goal is to develop drugs that are going to transform patients' lives. So just a real brief background on Ionis. We're not a major pharmaceutical company like you see on television all the time, but we are a biotech company located in San Diego County. We're about 30 years old, and our focus, and I'll get into this, is on RNA targeting therapy, and I'll explain what that is in my talk. We're roughly about 400 people as a company, so we're relatively small as a biotech. And really where we focus is in drug discovery. So we have a technology platform that we use to identify drugs that could be potential treatments for a variety of different diseases. And what our group does is develop those through early clinical stage programs, and then oftentimes we'll partner with large pharmaceutical companies, such as Biogen, who's our partner for Spinraza and a couple of the other products that I'll talk about today. And so one thing I do want to reinforce what John said is that doing biomedical research is really a consortium of a number of different people that participate in the activities. Where institutions like Stanford are really important for this triangle over here, where and it's not meant to be per size, that there's a lot of clinical research that goes on at Stanford, but a tremendous amount of basic research occurs in academic institutes like Stanford, and we as an industry are really heavily relying on that for our programs. And ultimately, as patients, you need that basic research. And I'll talk about a project that came out of Stanford that we're working on. It really is a basic research project. The next stage is translational research. So how do you translate these basic understandings in discoveries and start thinking about how do you make therapies out of them that are going to help patients? And again, that's where institutions like Stanford are phenomenal in doing translational research. Then ultimately in clinical research, how do you study drugs and study various aspects of the disease and get a better understanding of the disease? And again, institutes like Stanford are important. Where I fit in is in the pharmaceutical industry, and we do some basic research, but you see it's inverted. We do very little really truly basic research. Where we start getting involved is more in translational research over here. And then finally, as we're testing drugs going forward, a lot of clinical research that we use to help determine whether our drugs that we're developing are effective and safe for patients. It is really important to understand that we're really tied at the hip. We need the clinical research and the basic research that's occurring in institutions like Stanford, and without that, we would not be able to develop drugs. And then conversely, we have the expertise in how to translate some of these findings into novel therapies that we, again, work with the clinicians at institutions like Stanford to be able to go forward. So it really is a true ecosystem where science and biotechnology are working together. And this quote is from Dr. Zerhouni, who used to be the director of NIH a few years ago, that it is important that we recognize it's not, we don't work in isolation, that we work very closely together going forward. And so what I'd like to do is give some examples of, first off, introduce our technology, then give some examples of how we're using the technology to develop therapies for neuromuscular diseases. And one thing that has happened in my lifetime in science is the genetics of what causes a variety of different diseases has become very active area of research, and a lot of discoveries have been made. And so just to give you an idea, this is a little bit of a history lesson, is that there's a neuromuscular disease called spinal bulbar muscular atrophy. It's commonly called Kennedy's disease. The gene that causes that disease was discovered in 1991, so roughly 27 years ago. That was followed very rapidly by a number of discoveries of genes that cause disease. Huntington's disease, which is a neurological disease, was in 1993. The first gene that was identified that causes ALS was discovered also in 93. And then the gene that causes SMA, which Dr. Day just talked about, was discovered in 1995. And so, as patients, you may ask, why haven't we had drugs that will treat these diseases? And because we've known what causes them now for 25 years or more. And part of the issue is that science isn't that simple. Even though you know what causes the disease, how you develop therapies for treating these diseases isn't always obvious or easy to do. It's part of what makes drug discovery a very difficult enterprise. And just to highlight a couple of recent discoveries, the gene that causes FSHD was described in 2010. And then there's additional genes that have been described that are contributing to ALS have been in 2011. So there's been a lot of nice discoveries. And what I'm going to tell you in the next few slides is that we have a technology that can help translate these exciting scientific discoveries into potential therapies to help patients. And the way we do that is with a technology called antisense drugs, or antisense oligonucleotides. And That's been the focus of our company now for the past 30 years, is developing this technology so that we can bring therapies forward to patients. So what is Antisense? So I have to go back, or bring you back to some of the basic science. And so you know that the genes encode all the information in your body that tells your body how to function. And so one way to think about that is genes are the hard drive in your computer that really store that information. And that information is translated or transcribed into an intermediate molecule called RNA. And that RNA is then translated into a protein product. And most drugs today, such as the small molecule drugs, aspirin or antibodies or statins that lower cholesterol, all work on proteins. And so what we're developing is a new paradigm where we're trying to target this intermediate molecule, the RNA, and affect the production of a protein. And the way we do that is with a technology called antisense oligonucleotides. And so they're very similar from a chemistry perspective, very similar to natural RNA or DNA. And what we do is we bind to this RNA, and I'll show that on the next slide, and modulate the function of that RNA. So we either cause it to be degraded or we cause it to increase the production of a protein product. So it's we can kind of tailor which one of these processes you want to use based upon the chemistry and the molecular biology of what's known about the RNA. And so again, we bind to this RNA through what's called Watson-Crick base pairing. And so one of the beauties of antisense oligonucleotide technology is that if you know the genetic sequence of a gene, we can design a drug to bind to that gene just using some well-characterized binding rules that were discovered by Watson and Crick, in which they got the Nobel Prize for in the 1950s. And so if you have A binds to uracil, U, or C binds to G, et cetera. So knowing these binding rules, we can very easily design a drug that would bind to a target gene of interest and prevent it from being expressed. And so once it binds, as I said, there are a number of different mechanisms that we can exploit to either decrease the expression of a protein or increase the expression of the protein. And I won't go into detail, but this slide just kind of highlights a number of different mechanisms that we can do. And really what our company is focused on is learning how to use these mechanisms to develop therapies that may help patients with a variety of diseases. And one way that we have to do this is use chemistry, and I promise this is the only chemistry slide I'll show, is that that there are a number of different chemical modifications that we put into our drugs. And the reason we do this is that if you were to inject natural DNA or natural RNA into the body, it would disappear within 2 or 3 minutes. And so that's not really a drug. And so we use these chemical modifications to help in the distribution to cells within tissue, as well as be stable enough so that you don't have to inject or constantly infuse these drugs, that you can administer them infrequently. And so that's where all these chemistry modifications come to. Yes?
I think you have an error in the top right of the slide there. I'm just kidding.
Yeah. Very good. So this slide just shows, to give you an example of a number of different antisense drugs. They're not all our drugs, but a number of antisense drugs that are currently in development for treating neurological diseases. So nusinersen is shown at the top. That's an approved drug that's being marketed by Biogen for treating spinal muscular atrophy. There's another drug that's been approved for treating Duchenne's muscular dystrophy. These two drugs work by splicing modulations or to increase the production of a protein product. There's a number of drugs that are in development for treating another peripheral neuropathy due to a mutation of a protein called transthyretin. Those have been approved in the US just recently within the last couple of months. And then there's a number of drugs in development for Duchenne's muscular dystrophy, ALS, Huntington's disease, as well as some other neurological diseases that aren't shown on the slide. So it's a potentially a very exciting time for treating these neurological diseases. Using this technology platform, we've learned how to address some of the diseases using some of the genetic knowledge that was established 20, 25 years ago. We're now able to translate those findings into developing therapies that have the potential of treating the diseases. And so what I'd like to do now is just talk about some of the neurological diseases that we're working on. And one of the issues with antisense oligonucleotides is that they don't get into the brain when you inject them systemically or administer them orally. There's a barrier that prevents drugs from getting into the brain called the blood-brain barrier. And our drugs don't cross that blood-brain barrier. And so to get the drug into the brain where you need to treat a number of neurological diseases, we administer it into the fluid that surrounds the brain, the cerebral spinal fluid, using what's called a lumbar puncture. And so that's an injection into the lower part of the back. It's very similar to if you've ever had a spinal block for anesthesia. It's a very similar procedure. It sounds pretty traumatic, but it's being developed, and there's a long history of doing it so that it can be done fairly easily by experienced clinicians as a way to go forward. And so once we inject it into the brain, cerebral spinal fluid, what we found is our drugs distribute throughout the neural axis, so throughout the spinal cord and the brain. And so it gives us an opportunity to develop therapies that target a wide variety of neurological diseases. And I'll talk about a couple of examples. So the first example is a disease called spinal muscular atrophy. And so this is generally a pediatric disease, although there are adult forms of the disease as well. And it's the number one genetic cause of infant mortality today, or used to be. And with patients with the most severe form of the disease, the succumbing to the disease, usually by the second year of life. And so it really was a severe neurological disease. The disease is caused by a mutation in a gene called survival motor neuron, SMN1. And it's either due to a deletion of that gene or a mutation of that gene, so you no longer make this protein called survival motor neuron protein. It turns out that during our evolution, this region of the human chromosome was duplicated, and so there's actually two copies of this gene that differ by a few nucleotides or DNA molecules that differ. And one of the key differences is that there's a mutation or a change in a nucleotide that makes the SMN2 gene shown here produce a little bit of protein, which is sufficient to keep the children alive, but not sufficient to give them full function. And so as a technology platform, what we've done is identified a way that we can increase the production of this survival motor neuron protein. And it's basically a protein replacement therapy that we're replacing this protein that's missing using our genetic-based mechanism. And so what we've are doing is causing a change in the splicing, so it makes a protein product that essentially is identical to the missing protein in these patients. And so we've done a number of clinical studies, and some of the studies were done here at Stanford that range from the initial studies that we do are called phase 1 studies. They're focused on safety of the drug, so predominantly it's short-term treatment. A small number of patients get the drug in these phase 1 clinical trials. And from the phase 1 trial, you get some hints that the drug may have some activity or clinical benefit, but it really is, you have to be careful over-interpreting those. And then you launch, if the drug's safe and you can demonstrate that you have a path forward, you launch into what's called a phase 2 clinical trials. And we had 4 different clinical trials looking at different patient populations with the drug. And the goal of these phase 2 trials are, again, primarily safety. You want to look with longer-term treatment, is the drug safe? And then beginning to get some hints that the drug has some efficacy. And fortunately, with this drug, we were getting some pretty strong hints that the drug was efficacious. in these patients. And that was confirmed in what's called phase 3 clinical trials. And the goal of a phase 3 trial is really to focus on demonstrating clinical benefit. You again collect safety data because you're looking at a much larger patient population. But the goal is really to demonstrate clinical benefit that would support getting approval by the regulatory agencies throughout the world. And what we've observed in the SMA trials with Spinraza is that this plot is a number of different trials that we've plotted all in the same graph. And what we're measuring on this axis is changes in muscle function in these kids. So are they able to sit? Are they able to raise their arms, et cetera? And what we observed is that with treatment, we saw a marked increase in their muscle function. So we didn't just stop the progression of the disease, but we actually improved the symptoms in these patients. And what we also observed is that the blue line here shows the development if we started treating before they actually developed clinical symptoms. So early treatment actually had the most robust effect in these patients. Which is, again, very encouraging for other neuromuscular disorders, is that if we can use genetics to identify patients who are at risk for developing a disease and start treating them before they develop symptoms, that's probably going to have the most robust effect on these patients and may even prevent the development of the disease or minimally delay when the disease occurs in the patients. And so based on a number of all the clinical studies that I've showed, the FDA approved this drug about 2 years ago. And it's really the first drug that was approved for treatment of spinal muscular atrophy and is really transforming how we take care of these patients going forward. And with this pre-symptomatic dosing, it's helped drive putting SMA on a newborn screening so that eventually every person that's born, baby that's born, will have a test for SMA. And if they are positive for the gene, then they have an opportunity to go on SMA therapies. I will also mention that there are a number of other drugs that are in development for treating SMA that I think really are going to transform this disease going forward. It's making it a very treatable disease today. So in the next few minutes, I'd like to switch to some of the other projects that we're working on. They're earlier stages. So they're promising, but we haven't documented that they have the clinical benefit. But they're, in my opinion, very exciting as far as what the potential of the technology can bring. And so one thing that Dr. Day mentioned is that we're learning from our experience in SMA and applying that for treating other neuromuscular diseases such as ALS. And so today we have 3 programs in ALS with our partner Biogen. One that's targeting a familial form of ALS due to mutations in superoxide dismutase, and I'll go into that in a minute. Another one targeting the most recent genetic form of ALS due to mutations in the C9orf gene that I highlighted a few minutes ago. And finally, the work that actually originated here at Stanford identified a novel gene that may be targeted for treating all ALS or sporadic ALS, and that's ataxin-2. And that's earlier stage, but it's heading towards going into clinical trials in the near future. So first I'd like to talk about SOD1. So this is an ultra-rare form of ALS. It accounts for about 10% of all ALS patients. 2% of all ALS patients will have a mutation in the superoxide dismutase. We recognize that it's ultra-rare and it's not treating the broader patient population. But one of the reasons that we wanted to focus on this is it's known that this mutations in this gene cause the disease. And it's known that the cause of the disease is due to a gain of function, so this protein becomes toxic. And the thought is that if we could remove that protein from neurons where it's producing its toxicity, that patients may have a benefit. And it really gives us an opportunity to in a very careful manner, demonstrate that we're having a benefit in ALS patients by going after the genetic cause of the disease. And we can use that information then to learn how to apply this for other forms of ALS going forward. And so this was a program that we had taken into clinical trials early on. It was actually our first CNS-administered drug was to target this gene. And in the process, we did a phase 1 clinical study in patients. And what we identified is that with our new advances in the technology, that we could come up with a more potent drug. And so we went through an exercise where we rescreened, so we went back to the drawing board, if you will, as a way to to develop a new therapy for these patients. And what we identified is a drug that's 3 to 5-fold more potent than the original drug. And this is shown in this graph here, where this is I'm sorry, you can't see these, in the upper left. This is the original drug in the orange, and then the new drug is in the blue. And you see that it's more potent. It has a better effect on survival in these mouse models of ALS. And importantly, it not only prevented the denervation that you see in these animals, but it actually improved nerve function. And that's shown in the bottom part here. And it worked not only if we treated prophylactically before the animals developed symptoms, but actually after they developed symptoms, as you see in the lower right-hand panel, the blue line showing that we actually improved nerve function in the animals even after they were developing symptoms. So giving us some hope that, at least early on, some aspects of the ALS may be reversible before there's complete denervation. So this is currently in a phase 1 trial. We've completed with our partners Biogen, they've completed the single-dose study, which is shown on the left slide. And then we're currently in the multiple-dose study, and we should be in a position to announce data by the end of this year, beginning of next year. So things are going very well for this program, and I think, again, it gives us a lot of confidence that we will be able to advance this project forward into pivotal studies going forward. So just very quickly, we have another program on the genetic cause of ALS, and these were 2 seminal papers that were published in 2011 by 2 different research groups identifying a novel gene that called C9orf72, and for those of you who aren't familiar, that just means it's located on chromosome 9, and then there's sort of linear accounting on the chromosome 9. It's the 72nd gene on chromosome 9, so that's what that term means. But what it's done is really opened up, I think, a tremendous amount of knowledge about what can cause ALS based on the biology of this gene. It's caused by a expansion. So I mentioned that if you imagine that the nucleotides are letters, what happens in C9orf72 is you get this expansion of letters, or a stuttering, if you will, and that causes a number of pathologies or a number of side effects that contribute to ALS. This is about 8 to 10% of all ALS is due to mutations in this gene. And so we have a drug that we're developing to treat this form of ALS that just started clinical trials this month, and our hope is that will continue to advance towards developing a therapy for this form of ALS. And then finally, just to highlight work that really started as basic research, and there's a person at Stanford called Aaron Gittler who was studying ALS in flies. So it's a very basic research program. Looking at flies, Drosophila, fruit flies, as a model of ALS, and discovered that reducing this protein called ataxin-2 would help flies that had this ALS model survive longer. And then based upon some additional work, they identified that there are a proportion of ALS patients that have a mutation in this gene, again suggesting it could be an important gene for sporadic ALS or all ALS patients. And so this is a program that we're advancing towards clinical trials as well in the future. And then finally, just to identify that there are a number of other diseases that we're working on that range from Parkinson's, a neurodevelopmental disorder called Angelman syndrome, CMT1A is a program that we have active drug discovery programs, a number of ataxias, and then prion protein, GFAP for Alexander's disease, and then myotonic dystrophy is a program that we have had a clinical program on that was Stanford helped us do the clinical on on that. Had some encouraging data, but felt that we needed to go back to the drawing board again to develop a more potent drug, which we're in the process of doing, and hopefully that will go back into clinical trials soon. And so with that, just to highlight that at least from the data that we've seen today, that antisense technology does have utility for treating neurological diseases. And there's a broad spectrum of diseases that we're focusing on using this technology. We still have to prove that they're effective other than SMA. And so there's still a lot of work to be done. But it is something that we're focusing on as a company. And it's been a real pleasure helping to support these projects and develop therapies that are going to make a difference. And so finally, just to acknowledge all the many collaborators. This is a partial listing of the people that we've worked with to help develop these therapies. And the key message is it really is a consortium of advocacy groups that we've worked with, a consortium of academic investigators, and pharmaceutical companies that we've worked with to help bring these therapies forward. So with that, I'll stop. Thank you. Any questions or? Yeah, so the question is, does our technology have an ability to reverse the damage that's occurred in ALS? And what I could say is that that's what we're trying to figure out in the clinical trials today. We don't know the answer to that. It's, I think, possible, but we have to prove that, and we haven't done that yet. So it's something that we'll know in the next couple of years whether we can actually reverse some of the symptoms. There will be a point where you've lost so many neurons that we're not going to reverse that because you don't grow back motor neurons. But if they're sick and not working right, we might be able to reverse it earlier in the disease where they're sort of gain of function. Yeah. Got it. So I'm asked if you could write down your questions, and then do you want to do it at the end? Yeah. Okay, sorry, thanks. Yeah, so I guess we'll sorry, I didn't know that. With the panel? Or, I'm sorry. Right. Okay. So the instructions are that if you could write down your questions and raise your hand, somebody will come around and pick them up to be able to ask. Sorry. Yep.
A rare thing has happened. We are on time. And so you can take a 10-minute break if you need to, and then we'll get started with the two more presentations. Okay.
To Frank whenever we're
excited to have some people, like there are little cards, so you just write it down and we'll have somebody come around. Just raise your hand up. Perfect, thank you. Yeah, I have some in the back. Yeah, just making sure. Oh, whoops.
Oh, thank you. I'll take
Thanks
it
very
from
much.
you. I know. Yeah. Just, but I'll mention it.
Sorry about that. No, it's perfect. No worries. Thank you for helping with everything. I appreciate it so much. Of course. All right. And they'll bring it up in the back, so you're all set. It'll show up in a few minutes. I'll let them know. Are you ready? Or you want to take a few minutes?
I think we should get going. I think.
Okay, perfect.
Just so that we can be on time. Joe? I think we'll get started if that's okay. Thank you for coming back after the break. Okay. So thank you for coming back after the break. We didn't quite go through the couple of housekeeping issues, so sorry about the confusion a few minutes ago. If you get a chance, or if you have a question, we have two more speakers, including myself. Please write this down, or if you need help, one of us will come down and do the question for you if you like. One of the main reasons is that we want to be able to repeat the question here so that the AV folks can record the question and everybody can hear the question. And so we have two more talks, and then we have a whole half an hour for Q&A before lunch break. So that should be enough time to go through those things. And so my name is Sri Muppidi. I'm one of the neurologists who sees patients in the neuromuscular clinic and autonomic disorders clinic. And I see some of my patients in the audience. And I'll briefly talk about the immunological aspects of neuromuscular disease and how it might be relevant to even not typically thought to be a sort of a neuromuscular disease in terms of various things that Dr. Bennett already talked about. So here we go. So most of us learn something about immune system in high school and sort of college years. We all have immune system, and it's there for a purpose. It's there to fight against infections. It's there to fight against something foreign happening. There are two types of essentially immune system. There is a innate immune system, and there's an adaptive immune system. And the innate immune system is the one that works quickly, rapidly the moment you're exposed to any kind of pathogen. And that's the one that takes care of if you're exposed to a bug or a virus or so on and so forth, that's the one that takes care of it. And it has different components, different cells that take care of the immune system. And those of you who are on any kind of immunosuppressive drugs, physicians will sometimes check various lab tests to see how the different cell counts are. And we essentially actually check some of these cell counts to make sure that those counts are not too low, because that's one of the side effects of suppressing immune function is that you might be more susceptible to infection. But there is also an adaptive immune system. So if you're exposed to something and your body gradually builds up some resistance to it, and that has different cells, B cells and T cells. B cells will help you produce antibodies to help fight against it. T cells has a different function to fight against the infections. Even though we think of B cells and T cells and specific therapies which just attack the B cells, specific therapies which attack the T cells, in essence, they actually communicate with each other all the time. The whole system works completely normally in a complete homeostatic state, and nobody has any problem. But when something goes right, that's when the problem comes up. Or when we use strong drugs to attack either the B cell or the T cell, then you might have residual effects that we want to be able to anticipate. And we want to be able to attack those cells that are as specific to the disease causation as possible. Sometimes we can get to that level of specificity. A lot of the times we don't have drugs with that level of specificity. And so when we give drugs which work on these cells there is always this chance that we are attacking cells that are inherently needed to fight infection for other reasons, good reasons, and leaves patients susceptible for different complications. And that's something that we always think about when we use various drugs in autoimmune disorders. One of the questions that always comes up, why did I get it? You know, why did my sister not get it, my brother not get it? What did I do to get it? And the bigger question is we actually don't know. We actually don't know why one person gets an autoimmune disease. Most of the times we go back and think about, oh, you know, I did that, and did that trigger the process? In reality, very rarely do we ever find a specific cause and say, you know, you did this, or this happened, and that's it, boom, you got it. It's very rarely that simple. Most of the times, and I like this picture because I think it's an interplay, and not everybody's circles for the gene, for the sort of immune regulation, or for the environment are of the same size, just like it's shown in that cartoon. Some people have one circle that's much bigger, and some people might not have another circle that's as big or doesn't play as big of a role. But I suspect in most people who develop an autoimmune disease, these three things play some role. And if you hit that sweet spot, or if you're unfortunate enough to hit that sweet spot, then you might be more susceptible to developing certain autoimmune diseases. But I do think that there is probably role both in sort of genes that are difficult to change, but there might be a role with the environment, something that you're exposed to, for example. And there might be some inherent deficiencies, for example, that might change your immune regulation. So it's not a neuromuscular disease, but multiple sclerosis, one of the brain spine diseases. There's a lot of data that vitamin D plays a big role in multiple sclerosis. It's another autoimmune disease, so much so that vitamin D is levels are routinely checked and routinely replaced in anybody who has multiple sclerosis because of the large amount of data as the relevance of vitamin D in that particular disease. So with that, we'll go to some specific diseases. The best known one, the best known antibody-mediated neuromuscular disease is a condition called myasthenia gravis. It's an autoimmune disease that attacks the nerve-muscle junction where the nerve sends a signal to the muscle. It's well known because the antibody has been known for a long period of time. It generally manifests with multiple symptoms, but most common symptoms are patients have trouble with their eyes, so they might see double, or they might have droopiness of their eyelids. They might have weakness of their sort of tongue, swallowing muscles, or they might have a weakness of their proximal muscles, by which shoulders and hips. And so if you have symptoms of just your eyes, we call it ocular MG. If you have symptoms in other parts of the body, we call it generalized MG. Most of the times the diagnosis is fairly easy because there is a blood test, you can check for antibodies. Most of the times the antibody tends to be this antibody called acetylcholine receptor antibody in about 80% of the patients. If that's negative, there are a couple more antibodies called MuSK antibody or LRP4 antibody that can also be easily tested, again, blood tests. And if you have the antibody, if you have the clinical picture, diagnosis is not a problem. Rarely, we might need to do more testing for the diagnosis, and I'll go through that. Sometimes patients have significant weakness for a long period of time, and occasionally not every part of the weakness is reversible, even with treatment. So this is an example of somebody who has a mild eyelid droop, and you'll see on the sort of the B, she was asked to raise her eyebrows, and then she has weakness of her forehead muscles. Or she was asked to close her eyes, and she's not able to close all the way. And then you'll see that she has disease for a long period of time, so her tongue has become shrunk and smaller. If you have the antibody, as I said, the diagnosis is fairly easy. If you don't have the antibody, we have other testing. We stimulate the nerve, record the signals, and try to understand, is the nerve not talking to the muscle as well, or is there a problem with the way the nerve meets the muscle? A couple of ways we do that. So we stimulate a nerve and record signals. So on the left one, you'll see that the size of the response goes down as we keep stimulating the nerve. And that's because the signal is not going through and the muscle is getting weak and fatigued and tired. And the right one is a little bit more complex testing where we can record from individual muscle fibers and see if there is variability in how those muscle fibers fire, and that will also give you a sense that there is a problem with the nerve meeting the muscle. Just to give you a sense of those who have my senior have been to other support groups that I've spoken at probably have seen me draw this kind of picture. This is a complex picture, but the idea is that the top is the end of the nerve where the acetylcholine gets released and it joins the muscle and then increases sodium flow into the muscle and helps your muscle contract. This has happened billions of times since I've been standing here, so it's not a problem, right? Most of the times you don't have any difficulty with it. The problem occurs when you have antibodies attacking that receptor, that channel where the acetylcholine comes and joins it. You'll see these little red antibodies that are binding the receptors, either causing the receptor damage directly, or they're causing damage to the muscle membrane, or they're causing indirect damage because of complement and other autoimmune basis of damage. And what we do in practice in taking care of these patients is to directly address where the damage is. So either we increase the amount of acetylcholine by medications so that there is more acetylcholine to sort of get to the muscle, or we use various immune suppressive drugs on the left side of this side, which can attack different cells, either the B cells which produce the antibody, which T cells which are important for the immune basis of the disease, or we can do therapies like plasma exchange. We take out the antibodies, and recently newer drugs which can help with complement-mediated pathways in terms of damage to the neuromuscular junction. Most of the times, we can get the diagnosis fairly well. Most of the times, we can think about different drugs that might be optimal for a patient. One thing that I've sort of nationally tried to preach and at my local institute is to try to measure the disease the right way. Measure the disease the best way we can, measure the disease with the precision that we would use, for example, in the way Dr. Bennett talked about doing a clinical trial. There are lots of these outcome measures that are used in clinical trials that we also use in practice because it allows us to longitudinally look at a patient as we start treating them with any immunotherapy. We generally try to measure these things right from the beginning of the disease and then longitudinally follow them. So on the left bottom column, that's from a patient of mine. You will see the number was quite high. Over a period of time, the number came down. And then there was sort of relapse of the disease. The effect of the drug had worn off. And then you will see a little spike there. And then you give one more therapy, and then it's come down. And this is feasible. It doesn't take more time than usual clinic visit, but it provides some extra sort of granular piece of data that I think is relevant for day-to-day management of these diseases. Also, although I don't encourage that everybody do this, but there are some online ways where you can actually monitor your own disease based on your symptom burden. It's not something that I would encourage you to do every day because that's just not practical, but maybe every couple of months or so that if your doctor has put you on an immune therapy, you can actually monitor it over a period of time, and you and your doctor can look at the data together and see what kind of relevance that particular measure applies to you for your own daily living. These are fairly simple things to do. These are all validated measures. These are not measures that are just came up without any sort of background science behind them. And so they're really valuable that way. The next topic is inflammatory muscle diseases. So these are muscle diseases where there is a autoimmune basis as to why the muscles are weak or the muscles are damaged. We use the word myositis. Itis generally suggests some kind of inflammation. So we use the word myositis as in to suggest muscle inflammation. There are lots of different muscle inflammatory disorders. So there is polymyositis, which essentially means multiple muscles are involved. Dermatomyositis, which is skin and muscle involved. Inclusion body myositis, one of the muscle diseases which is much more difficult to treat. Necrotizing myopathy, which generally refers to the significant muscle damage. And there are some patients who have myositis in the context of having other connective tissue diseases, such as rheumatoid arthritis, systemic sclerosis. They have other lupus, other autoimmune diseases. We used to think of muscle diseases based on what a patient came with, what symptoms that they had. But there is a thought that the muscle diseases should be based on what antibodies they have. The point is that different diseases on the left column are caused by similar antigens on the right side. And there might be some basis to treating patients differently based on what might be the underlying antigen, because there is some data that if you have an antigen, let's say you have dermatomyositis, but not every patient with a dermatomyositis will behave the same way, will respond the same way, will have the same problems related to weakness or skin changes. There might be a role as to what exactly the antibody that they have that's causing the disease. So in practice, we try to measure or at least check for these antibodies at the beginning of the disease so that we can taper the therapy towards that particular patient. So as I said, most patients will have weakness. Some patients will have different kinds of skin changes. Patients with IBM tend to have difficulty sort of making a fist, so that's what that picture at the bottom is showing. We're not able to make a complete fist. And also they have significant atrophy of their muscle in the thigh muscles area. The diagnosis, again, is should not be that difficult because there are multiple tests that you could do to confirm. You can check a muscle enzyme level, which can go up in lots of different diseases, but you can check a muscle enzyme level. That will help you understand a muscle disease. We can do a EMG nerve conduction study, that's where we stimulate your nerves or record from your muscle and then try to understand if there's a muscle disease. You can do a muscle MRI and as that arrow points out, look at the changes in the muscle fiber that can also help you find out if somebody has a muscle disease. You can check antibodies, and that can also help you understand if somebody has a muscle disease. Or you could do a muscle biopsy. And John had earlier mentioned how he likes sample for everybody, for everything. And so we can definitely get a muscle biopsy. And we can get a muscle biopsy, and we can work with our pathologists, look at the muscle biopsy slides, and try to understand what kind of inflammation there is in the muscle biopsy. Is the muscle biopsy suggest to a polymyositis, a dermatomyositis, or inclusion body myositis? And the muscle biopsies have different signatures, and so that will also help us understand the degree of the muscle disease and extent of the muscle disease, and if we can narrow it down to one of these specific types. And there are different immune therapies available for all of these conditions. A lot of it is tapered, just like in myasthenia, to what the disease that you have and how severe it is, and what might be the best first agent, and followed, and so on and so forth. And the last section that I was going to briefly talk about is neuropathy. So these are inflammatory neuropathies, neuropathies where there is an underlying autoimmune basis to it. Neurologists love their alphabet soups, and so that's why you will see all these little acronyms on the left side. But they all essentially indicate different types of autoimmune diseases affecting different parts of the nerve. Either it's affecting the motor nerve, as in related to strength, or sensation nerve, or a combination of them. And then the D generally stands for demyelinating, so which is the outer myelin sheath on the outside of the nerve. A is generally axon, which is the one inside. There are different types of inflammatory muscle diseases, just like different types of Charcot-Marie-Tooth, one of the nerve diseases, genetic nerve diseases. And the way we can diagnose them, we can examine patients, and they have sort of a unique pattern of weakness with sensation loss or reflex loss. We can do that. We can diagnose them based on electrophysiological testing. Again, we can stimulate their nerves, record signals. And because myelin is important for how fast your nerve conducts, we can actually see different responses that change if the myelin is much more affected versus the inside part. The axon is much more affected. Or sometimes we could do a biopsy. We can take a sample of your nerve and look for specific changes that will help us understand is the myelin is more affected or the axon is more affected. The diagnosis also sometimes involves spinal fluid testing, different lab testing looking for an underlying sort of explanation for the disease. One thing that has come up in the field of the CIDP and other autoimmune diseases is these newer antibodies that were discovered in the last few years. These were not known before, and they probably explain why patients with CIDP, one of the inflammatory demyelinated neuropathies, not everybody behaves the same way. Some patients have severe weakness, some only have mild weakness, some have a lot of strength issues, some have a lot more sensation issues. And the thought is that these antibodies bind to the nerve at specific areas called nodes of Ranvier, and that's where one of the neurologists suggested we start calling them nodopathies because it's affecting the node, not the whole nerve. And they might lead to sort of specific therapies, or they might require specific therapies, unlike all patients with CIDP, because they have an underlying genetic basis that affects what happens to these patients and how their disease progresses. And so I think that's where the field is going to. I suspect going forward with all three diseases, we'll sort of reach a level of granular specificity where we not only say you have CIDP or you have myasthenia or you have myositis of one type. Hopefully we can say more about the disease that one has and then taper or titrate the therapy specific for that particular treatment. Now, some of you, because I see only some familiar faces, so not everybody here has myasthenia or one of these three diseases. And so you might say, well, I don't have one of these three things or these groups of diseases, and so the immune system is not a problem for me. I'm fine. Actually, I disagree. I think John sort of mentioned that at the beginning. Even for diseases where we know there is a primary genetic basis for it, either we use immune therapies to treat some of them. So for example, we use glucocorticoids, steroids to treat patients with Duchenne muscular dystrophy has been the therapy for a long period of time. And I suspect will continue to be the therapy even as the newer drugs start to come into play. So there is a definitive role there. We also sometimes use immune therapy when we are treating patients with known genetic diseases. So for example, there is a disease called Pompe disease where there is a deficiency of an enzyme which helps you use glucose in your muscle. And it can affect young people and sort of later in life. And in some patients, when they get the enzyme replacement, they really react adversely to it because their body thinks that the enzyme is foreign and start attacking the whole process. And so we actually use same mechanisms of sort of immune tolerance and how best to manage that adverse reaction so that the enzyme has a great opportunity to make a difference to the patient. So I think there is a role beyond the classically defined neuromuscular diseases which are immune mediated. I think the field is moving in such a way that there's probably going to be more and more therapy options just for those immune mediated diseases, and there's going to be a strong interlay in when we have newer genetic therapies coming up. If they have or if they produce an autoimmune reaction, it might still be relevant, and we still might have to use drugs or therapies to help treat those side effects, or prevent side effects, or make sure those drugs have great opportunity to help you. So I'll stop there, and then we'll take questions right at the end. Okay. Good. Thank you. Thank you. Yes. Sure. 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 So I'm going to introduce Tina.
Okay, sounds good. Okay, hi everyone. Thank you so much for being here. I'm really, really excited to be able to talk to you today about a topic that's very near and dear to my heart, and now I get a platform to talk about it for 20 minutes. As we were looking through the program and trying to, this is a very broad meeting talking about neuromuscular disease, what topic really affects every one of you here? And so one of the topics that popped into mind was muscle weakness and muscle tightness or contracture development. So anyone in the room here who has a neuromuscular disease may have some muscle weakness, and that muscle weakness may contribute to some tightness or contractures. And how does that actually play into function on an everyday basis? I'm gonna wait for the slide deck to come so you don't just hear me rambling. But my name is Tina Duong. I'm a physical therapist here at the Clinical Neuromuscular Program at Stanford.
So it's other guy's slides. Dr. Muppidi, you have to open yours really quick.
Okay. I'm not a PC person, so I'm going to need help.
Right there.
Is this one?
Yeah.
Okay. And maybe? All right, great, we can get started. So just to kind of piggyback on what's been happening and all the really exciting times in drug development for neuromuscular disease, as highlighted by Dr. Bennett earlier, my job here today is really to talk about a topic, as my previous boss said when I wanted to do research. He said, what do you want to research? And I said, you know, contracture development. He looks at me and thinks, you know, that's not really a cool topic. And it's not a cool topic because I'm not going to fix anything. I'm not going to tell you a cure for contracture development or anything like that. But it is a problem that when we're in clinic as physical therapists and occupational therapists, you ask us every day, how do you manage the contractures to move better? And so this is relevant in clinic, but it's also relevant in research, because as drugs are being developed, how do you prime your body and keep it as healthy as possible so whenever a drug is available, your body is in the best condition it can be to reap the best benefits. And so today I'm just going to go through how all of this interplay together. So when you're looking at the musculoskeletal system, you have muscle, and it's dependent on the muscle length, and the skeleton system, which affects the alignment. So as you can imagine, if you have any contractures or shortening of the muscle, it's going to affect how that's pulled on your skeletal system. And so altogether, that will ultimately affect your muscle function and activity. And so just a very broad overview. So today, these are the primary things I'm going to talk about. The bone. That's the skeleton that the muscle is going to pull upon, but there's two parts of the muscular tendinous region that we're going to focus on when you're stretching. So when you're stretching, you're going to either stretch the muscle belly, and the muscle belly is surrounded by fascia and then smaller myofibrils. And all of those things are stretched whenever you do any kind of stretch. But in addition to that, you also have tendons. And so the tendon is the white part that connects, I don't know if you can see nope, you can't see my pointer, that connects the muscle to the bone. And so when things shorten up, there could be shortening of the muscle fiber and also possibly shortening up of the tendon. And so what we don't know is that when shortening happens, where is that actually happening? But what we do know is when it does shorten, it definitely affects your function. And so, in general, a very generic term for shortening of muscles is contractures. And that can happen in multiple ways within multiple regions of the body, including the muscles, the skin, connective tissue, and basically it means some kind of restriction to range of motion. And for individuals like yourself with neuromuscular disease, any kind of imbalance or muscle weakness around a joint may lead to some kind of tightness or contracture development. And this could be based on the disease, it could be based on the positioning, or just the inability to move the muscle to move it throughout the joint. And so, in general, in contracture management, we have a few purposes in why we recommend this. So every time you go see one of the therapists, we'll talk about you should do stretching. What are your stretching exercises? What are other mechanisms for you to manage it? Some of the purposes are to maintain the muscle length so that you can function well. Some of it's to prevent some of the orthopedic deformities from not being able to move the muscle through the range. Other times it could be because to decrease pain, and then ultimately to improve function. So all of this together, our goal is to make you be as independent as possible and best address the quality of movement. And so in contracture development, broadly speaking, for individuals with neuromuscular disease, this is just a very small group of muscles that may get tight. And so generally, a lot of you have ankle dorsiflexion tightness, and then others may have iliotibial band tightness. For those who are in a wheelchair or sitting more and starting to walk less, you'll have hip flexor tightness. And hip flexors are the muscles that are attached from the back of your spine to the front of your hip. And so because of that, what comes along with hip flexor tightness may also be low back pain. And then, of course, along with those of you who are sitting in chairs more, you may have knee flexor tightness that will ultimately affect your ability to stand, making it harder. And then some of the more distal muscles are the wrist flexors, which is at the front of the palm, and then elbow flexors, which may affect your ability to groom yourself or eat. And then so how does this affect function? And so biomechanically, what happens when you have this shortening is that you have changes in torque of the length tension curve between the muscle and its connection to the bone. And because of that, you may have reduced power or velocity that's going to be required for you to move well or to even get your perform some of your activities of daily living. And so that may include things such as getting more difficulty getting up from the floor, getting up from a chair, climbing stairs, etc. And so how does contracture and function really relate to anything? Those of you in clinical trials, you'll notice that there's inclusion and exclusion criteria. And sometimes those inclusion and exclusion criteria have functional definitions, and they do come out of somewhere. And so when we're studying a drug or a therapy, you want to make sure that the study population that you're studying is the same or homogeneous. And so if someone is really, really contracted, even if a drug is going to make them into Superman, they're not going to, if they have a contracture that's so tight, they may not be able to show those functional changes. And so that's why when you see some of these inclusion and exclusion criteria, you wonder why they're being so restrictive. This may be some of the reasons why. And then on top of it, the effectiveness of treatment. So, like I said, if your joints are contracted and you gain strength, but you're limited by the contracture itself, are you actually going to see the functional changes that's part of the clinical outcomes or endpoints that's being studied by the research trials itself? For those of you who are on Spinraza, as you probably have gone through an hour and a half of evaluations with us, you'll start to notice as you're telling us your what's changing in your body, we try to find ways to measure it better. And that's kind of a moving target right now as therapies are coming about. And so that being said, I want to give an example of how range of motion really does affect function and how that's part of a decision making in clinical trials. So I've been part of a group that has looked at Duchenne muscular dystrophy for the past 7-8 years. And this trial's been running for 7 years or so. It's an international trial with over 300 patients. And we looked at a lot of data, but the one I want to focus on today is looking at strength and range of motion. And so as many of you who are familiar with Duchenne, typically the time velocity tests have been part of an inclusion and exclusion criteria. And in some trials, so is range of motion. And there is actually method to the madness as to why they ask you to do that at screening. It's because the natural history is showing that there are predictors of when you may lose function, you as in the person with Duchenne. And so on the left hand, you'll see that there's the 3 outcomes, which is the 10-meter run walk, the ability to climb stairs, and standing up from the ground. So many of you are very familiar with that. You get, when you see your clinician, they have you do that every time. And that's because these time function tests are possible predictors of the loss of an ability. So here on the top hand, you'll see that there's knee extension, ankle range of motion, age, and steroid use. These are the variables that may contribute to how fast you can perform those outcomes. And so as you can see here, where the variables we picked were the ones that we thought may actually affect that velocity, and it's actually very true. All of these variables, including ankle range of motion, is a significant factor as to how fast you can walk, run, or climb stairs. And so that being said, what are some of the approaches to managing contractures? And so there's conservative approaches, which I'll talk about today, and then there's surgical, that is not my expertise. I don't come, I tell all the kids I don't come with a knife or needles. And so we'll talk about short and long duration stretching. And the reason why I define that, because there's different methodologies in how we're going to approach it. The short duration stretches really is anything less than 30 minutes. It's your manual stretches that you or your caregivers performing with you, or an active stretch in which you're performing yourself. And then there are other methods where there's straps and standing frames that are recommended by your therapist to get better alignment and some stretching from your system. And then the long duration stretching, you'll notice that you get recommendations for splinting, orthotics, nighttime splints, and the intent of that is to hold that elongated position for a longer period of time. Because some of the animal research, or the mice-rat research, shows that if you hold it for a longer period of time, there's increased likelihood of increasing that muscle length itself. So then when we're talking about modes of stretching, there's all kinds of stretching, and stretching has been very, very thoroughly studied in sports medicine. So we learn a lot from sports medicine there. But to tell you the truth, what they come up with at the end of the day is they don't know what the best stretch is. Because there's different types of frequency, duration, speed that's required that could change or affect how well a stretch may be effective. So that being said, how do you make decisions on what to do? So we want to start with some basic understanding of the pathology. So basically, your diagnosis, because your diagnosis is going to affect how your body responds to the stretch itself. And so if you have Duchenne, for example, and you have increased fibrosis, it affects connective tissue, you're going to have less extensibility in your muscles. And so when you're stretching, you're not getting that change as much as you would for someone who has normal, more normal muscle. And then based on the pathology, we want to understand what the physiology of stretching is. And so we talk about stretching in very broad terms, but what is it actually doing to your muscle fibers itself? And then that'll lead to some of the decision making on management. And so there are a few things to consider whenever you're stretching. And so when you're stretching, the question is, are you lengthening the muscle, or you're lengthening the tendon? Because the quality of the stretch really matters. If you just pull on a limb, are you actually really stretching what you need to stretch? If the tightness is coming from a tendon, you really have to focus on the musculotendinous piece of the muscle, so you're not just pulling on the muscle belly. And a good example of that is the foot. So I'm going to pretend my hand's the foot, and you have the plantar fascia down here. And so a lot of times whenever someone stretches this, what you're really wanting to stretch when you have tightness is the Achilles tendon. So that it's very, very specific. But the problem is when the Achilles tendon is being stretched, a lot of times people are stretching the toes. And so what happens when you stretch the toes and not focusing on the Achilles tendon, you make kind of what they call a rocker bottom foot. You overstretch the plantar fascia, which doesn't put your foot in the best position, and you're not actually stretching the Achilles tendon. So it is the devil in the details. And so when you're stretching, that kind of focus really does matter on the effectiveness of the time that you're spending to do it. And then also, a lot of literature talks about stretch tolerance. And what that basically means is your tolerance to stretch. And the literature is pretty firm in this. There is increased range of motion when you do stretching. And the reason that's the case is a lot of times you just tolerate it better. You have less pain associated with stretching, especially for individuals who don't move a lot. So if you don't move a lot, your fascia is tight around your muscles, and so especially with kids, if they're stretching and saying it's painful, what is the actual pain? Is it the pain of just movement and lubrication of the limbs that they're not used to, and you have to do it slowly, or is it actual pain? And so a lot of the studies out there in sports medicine show that increase in range of motion is highly, highly associated with just decreasing sensitivity to the stretch itself. And I want to highlight what Dr. Magnuson from Denmark, he has done a lot of research in stretch, and he looks at muscle extensibility. So we're going to talk about that in a little bit more detail. But when you go to clinic and you're performing all these functional tests, there is only one way besides looking at your function to really measure how long your muscles are. And it's kind of rudimentary, to tell you the truth. It's this goniometer. It's this plastic measuring device, and they pull your limb out as far as it'll go and measure it. Does that tell you anything about whether they're measuring what aspect of tightness is in there? Is the tightness in the joint? Is the tightness in the muscle belly, in the tendon? We don't know. And so that being said, as I present you some of the literature that's out there in stretching, you'll understand why there's some discrepancy on the ability to prove its effectiveness or not. So that being said, muscle tension. So muscle tension is really how much tone you have in your muscles. And so there's active tension and there's passive tension. So active tension is your basic muscle tone. What we're going to talk about with stretching is passive tension. And that has to do with the viscoelasticity of your muscles as well as the surrounding fascia. So there's a study in the 90s that they did in rabbit and mice soleus, and the reason they do that is because they can pull these tendons out of animals, and we just don't do that with people, which is a good thing. And so what they found is in an immobilized muscle, their question was, what shortened first? Was it the muscle belly, the muscle fibers, or the tendon itself? So you could see on this curve here, the change in length being on the x-axis at the bottom, and the tension that's on the y-axis at the top. T stands for tendon, M stands for muscle. You see that the tendon is going to be the first thing that makes the change. So the first thing that shortens up are the pieces of the tendon, and followed by the muscle itself. So as we move forward, I know there's a lot of differences. Human muscle is not the same as mice muscle, but this gives us some insight on what immobilized muscles are reacting to whenever they don't get that change. So in stretching, there's two phases of stretching. There's a dynamic phase and there's a static phase. The dynamic phase is basically the more transient stage, okay? And I'll go into a little bit more detail about that phase in a little bit. But the static phase is the one that we focus on when we get to the end range of motion and we hold it, because what we really want is to see some kind of adaptation to the muscle, to the physiology of the fibers, so that we can actually elongate it to improve our quality of movement or range of motion. And so Magnuson's work really have shown us that if they're in these two phases, you'll see here up at in the first phase, or the dynamic phase, what you're doing is you're just taking up the slack. And once you take up the slack, what happens is there should be a viscoelastic response where you put the same amount of force will result in a sustained elongation or relaxation of the muscle fibers itself. And him and his lab have found a really good way to measure that, one which we weren't able to before. And so that gives us some insight on what the muscle is doing. And so when I apologize, I did this on a Mac and it translates really weird into a PC, so there are mistakes on my slide. So when you go to clinic and they tell you when you're stretching, stretch slow and hold it for a certain period of time, there is also a method to that madness, or at least the concept to us understand the physiology of stretching. So the reason why we asked you to stretch it slow is because when you're first stretching it, there are things in your muscles called Golgi tendon organs and Pacinian corpuscles that are basically sensing that movement. So it's saying something is happening here. And then the muscle spindles in your muscle fibers are recording this information and sending it up the spine to your brain saying, oh my gosh, somebody's stretching, let's quickly protect ourselves and contract. That's the stretch reflex. And so you want to stretch slow because you want to dampen that stretch reflex, because right when that happens, almost simultaneously, the lengthening reaction is going to react to that. And that's going to dampen the response and allow your body to chill out and allow that stretch to happen. So that's the reason why we recommend the stretch being slow and held for a prolonged period of time. That being said, I want to also highlight work that's done by Magnuson in the 90s, actually. He looked at stretching in a lot of different groups, and what he finally found is that 90-second stretch seems to be the ideal time. And he looked at stretching in a prolonged 90-second period, as well as 3 segments of 30-second repetitions with maybe 20 seconds in between. And he wanted to see if there's a difference in the totality of 90 seconds in short duration versus that sustained long duration, if there's any differences in the amount of tension that's in the muscle or the relaxation. And what he found is that 90 seconds is the ideal length of time to decrease the amount of pressure that's in the muscle when you're stretching, so it allows for a better stretch. And there wasn't a difference between 90 seconds sustained or 30-second periods of equaling up to 90 seconds. And then, so other research out there that's in neuromuscular disease about stretching, there's a study that's recently published in 2016 that looked at serial casting, orthosis, stretching, and different other standing devices. And so the population they studied were cerebral palsy, Charcot-Marie-Tooth, and Duchenne. And what they found is that there's really not enough evidence to support passive range of motion or positioning for prevention of contractures, and there's basically weak evidence. And so, you read this, and we're telling you to do this all the time. It's really disconcerting, right? And then on top of it, there's a systematic Cochrane review, and those are studies that look at multiple studies about stretching or about a certain topic, and then they review it. And so this study found that they even made harsher statements, such as there is no evidence whatsoever that stretching is effective in contracture management. But all that being said, when you're stretching, that's not all that you're doing it for. Right? There's benefits to stretching besides actual range of motion. And like I said before, we don't even have the proper measurement techniques or devices to really measure what we think we may be stretching. So there's, in stretching, the benefits include increased blood flow to the muscles. As we all know, if we're sitting for a really long time and we are not getting up and down, we feel stiff and we feel some pain. So we need blood flow to the muscles. And then also as you move through stretching, you're lubricating the joints and the articular surfaces. That's really essential in joint and bone health. And then muscle stiffness in general. We know in the sports literature that muscle stiffness leads to muscle injury when you're moving. So you want to keep that extensible, and that is well documented in Magnuson's work, that if you stretch for 90-second periods, that you do have that extensibility. And then, as we know, with stretching, collagen is a big factor in it. And collagen has shown that if you move more with ease because it affects collagen itself. And so basically, the stretch recommendation is that you stretch for 90 seconds to increase the extensibility with a rest, with some breaks of at least 20 to 30 seconds in between, or a sustained 90 seconds. And so the take home here is that just because the literature out there is saying that stretching and stretching short or long duration is not necessarily proven to manage range of motion, it does not mean it doesn't have its benefits. So contractures do have effect on muscle force production, your quality of movement, management of pain and stiffness. And prevention is key. So as we're looking forward to future therapies, we want to make sure that we are supporting you in making sure your musculoskeletal health is in the best condition, so that as drug therapies are coming down the pike, that you're in the optimal shape to receive it. And with that, I thank you for your attention.
So if we could just announce if everyone could just write down their questions and raise their hand, and we'll have a panel. I'll make some program notes.
So if everyone can write down their questions, and if you have questions, please raise your hand, and someone from the Stanford Neuromuscular team will pick it up, and we will do a Q&A.
Hi. Whoa. Sorry, I'm Jennifer. I'm the Community Outreach Liaison for the Neuromuscular Program. So while you're writing your questions down, I'm going to ask our speakers to come back up to sit on the panel, because you guys will be answering the questions. And while we're waiting for questions and our panelists to come up, I'm going to give you a couple program notes. So we will be serving lunch outside, and you'll pick up your lunch. There'll be Stanford staffers that will hand you your lunch box, and you're welcome to sit outside or inside, wherever you would like. There was the question of whether or not we are videotaping, and we are taping. We have a YouTube channel, and if you registered, you should be getting my emails, and there's hyperlinks in the emails to our YouTube channel. I will email everybody and let you know when the footage from today is uploaded to YouTube. Also, for the breakout sessions, just so you know, the motor neuron ALS and SMA breakout session will be here in McCaw Hall. So after lunch, the breakout sessions start. And if you are interested in the ALS and motor neuron breakout with Dr. So, just stay in here. Otherwise, all of the other breakout sessions are on the opposite side of the building. So you just keep walking until you dead end into the Fisher Conference Center. And there's signage for each breakout session outside the room. So the program is a little bit misleading, so just check the signage. And are we all ready? Okay.
Stay there for a second.
Okay.
I got something to say.
What's that? Okay. Does anyone have any questions that someone didn't grab? You can write questions as the speakers are speaking, so don't worry, just raise your hand and we'll have someone come grab a question.
I'm just going to ask Jennifer to stand for a second, because those if you have dealt with group of physicians, you know how difficult it is to get them in one place and to organize a meeting like this. So if you could just give a big round of applause to Jennifer and Joe.
Yeah, thanks to Joe.
Because without them, I don't think this will run. So I'm going to introduce to one of our senior faculty, Dr. So. So he's going to quickly go through the questions, club them, and then we'll try to answer them as many as we can.
All right, there are a lot of questions here. I'll try to lump them, and also for the sake of time, I'll get our speakers start working on it. So let's start with Tina while you're still warm, so already warmed up. This is a question of, can overstretching or continual stretching consistently be harmful for muscles?
So that's a really good question. The answer is we actually don't know. And because of that, we actually convened a meeting in March, and the NIH has been very interested in this as well, to understand what stretching is doing to muscle. And so there has not been any studies in MRI or anything to actually understand when you're stretching, especially with a neuromuscular disease, what is being stretched and what is not being stretched. And so our recommendation coming out of that meeting is that when you're stretching, to not over push the stretch, especially if you have neuromuscular disease, or if you know your diagnosis is a dystrophinopathy or something that affects the sarcolemma or the membrane stability of the muscle itself, because we don't know how much aggressive stretching is, whether it's causing damage or not. We haven't seen that to be the case, but from a imaging perspective, we don't know that information.
It's clear that we won't be able to answer the vast majority of the questions, but let's keep going. Before you leave the topic of stretching, One question is stretch how many days a week?
How
Quick
many-
answer.
How many days a week? So as I said before, stretching doesn't necessarily prevent contractures from happening or increase range of motion. So prevention is key. So for any of us, the recommendation for every single one of you, whether you have neuromuscular disease or not, should be daily. And so if you stretch, you're able to move better. So just think about stretching increases the extensibility of the muscle. So if you stretch, it puts the muscle at a more optimal state to have a better muscle force contraction. So the stretching is very, very beneficial for normal muscle, but even probably more so for those with neuromuscular disease.
This is a question for Dr. Bennett. Antibodies to Spinraza or nusinersen has been reported, but does not appear to affect treatment. Any further comment? Can you elaborate?
Yeah, so I don't want to speak specific for Spinraza because that's really a Biogen program, and I'm not as intimate with what's being developed in the commercial setting. But I can speak more generally about antibodies to antisense drugs, and we have seen in some patients that they'll develop an antibody to the drug. It has not affected either safety or efficacy in our other programs, where we haven't had any adverse effects that we've detected because of people developing an antibody to the drug, nor has it impacted the efficacy of the drug. So a lot of times when you develop an antibody to a drug, it prevents it from binding to its receptor. We haven't seen that so far, and so I think in both cases it's an immunological observation, but it hasn't really translated to a clinically meaningful outcome.
Another question here is asking for elaborations on myotonic dystrophy. I think you mentioned that in your talk.
Yeah. So as many of you know, we had a study where we were testing an antisense drug for type 1 myotonic dystrophy, and Stanford was one of the study sites for that clinical trial. What we observed in that study, for those of you who may have participated, you know you got a muscle biopsy that was part of the study. And that muscle biopsy allowed us to do two things. One was to measure the drug concentration in skeletal muscle, which gave us some context to whether we were getting enough drug in muscle. And then also we looked for drug effects of the drug in skeletal muscle. And with myotonic dystrophy, there are a number of molecular changes that we could detect in the RNA that's being produced in the muscle called splicing. And so there's different splicing changes that we could detect. And so in the study, what we observed were that the drug levels that we were getting in muscle was much lower than, or I should say not much, but lower than what we anticipated we would get based upon the mouse and monkey data. And we felt that the drug had some evidence that it was doing what it should be doing, but the doses that we're going to have to go to would have been too high. And so what we're doing now is screening for a more potent drug to bring forward back into clinical trials so that it'll make it much more convenient for the patients to get the drug.
Thank you. A question for Dr. Muppidi. In myasthenia gravis, I believe you mentioned that weakness can become fixed. Can you expand on that?
So sometimes, and doesn't always happen, but certain muscle groups, so muscle groups for eyes, for example, if they are not treated adequately for a long period of time, or even in spite of being treatment, I had shown this cartoon of where the nerve signal goes to the muscle, and if you have damage to the receptors on the muscle side over a long period of time, the muscle membrane becomes smoother. And even if you are getting more immune therapy or specific therapy, whereas at the beginning we all have a convoluted muscle membrane where the nerve meets the muscle, and if it becomes smooth, it doesn't work at the same level of efficiency. And so we call it end plate myopathy. And occasionally, we see that in patients who've had myasthenia for a long period of time. There's no specific way to treat it apart from trying to treat the best way we can and trying to treat all symptoms that the patient has in the best way we can. There was one other question I had here that I think is relevant to a lot of the things that came up today. The question basically says, John Day, that inflammation is part of all neuromuscular diseases. So do certain medication like ibuprofen, are they better options than Tylenol? My personal experience has been that patients find more benefit from ibuprofen, but there are side effects from taking ibuprofen or similar NSAID medication for a long period of time compared to Tylenol, so they need to be sort of weighed against each other.
This is probably a number of questions directed to Frank, to Dr. Bennett, asking for further information about developments for treatment in FSHD, Kennedy's disease, and CMT1. How much can you spill the beans?
Yeah, so we have over 30 different research programs where we're trying to identify therapies for these inherited disorders, including FSHD, Kennedy's disease, and CMT1A. They're currently in what we call drug screening, so we're screening both cell culture as well as in mice, trying to find the optimal drug to bring forward. That generally takes a few years to do. The issue with both CMT1A and FSHD is that they're in target tissues that are outside the brain. And so with CMT1A for those is Charcot-Marie Tooth disease, which is a neuropathy, so you're trying to target Schwann cells in the peripheral nerves. And we have to figure out how to get the drug into those specific cells to have the activity. That's a little bit more challenging than it has been for delivering into the brain. But we're working on it, and it's a very active program. Same thing for FSHD. Based upon our experience with myotonic dystrophy, we identified that we need to do some additional chemistry modifications to get better uptake into muscle, which is what you'd want to treat for FSHD. And so that's part of the program, and it's just something that we're working on today to actively try to find drugs for these diseases.
This is a question for everyone here. What do you think of CBD and marijuana in neuromuscular diseases?
So it's legal in California. There are physicians I know who prescribe it. I don't think anybody at Stanford prescribes it actively. AAN, American Academy of Neurology, has some recommendations for different types of pain syndromes. And I think there is some data that it does help with pain. But I'm not aware of any specific data in terms of actually changing the underlying physiology or the pathology in different muscle groups. I don't know if it makes a difference in terms of stretching or relaxation, or I'm not sure.
Yeah, and of course there are not studies in physical therapy or in any rehab that looks at CBD or marijuana, but just anecdotally, patients who use CBD or marijuana have stated decreased pain in their improved tolerance to stretching, for example, as part of that, what Dr. Muppidi said.
Similar vein of thinking. This is a question about steroid. Steroid can reduce inflammation. How does it help is one question. And the question posed to also say, well, in people without some of these diseases, inflammation is a natural part of muscle regeneration process. I guess the question is, how do you reconcile all that?
Right. So yeah, that's a great question, and that's so at least in the autoimmune diseases, steroids helps by controlling some of the factors which are actually causing the damage. The concern is that even in non-autoimmune diseases where the inflammation is a secondary process, sometimes that secondary process can have such a large overdrive in terms of the damage to the muscle that it's not allowing you to recover, even though it's a secondary process. And so when we use steroids for some of these diseases, non-immune mediated diseases, we're not trying to stop the natural sort of reflex healing that's happening. We're only trying to prevent further damage that we think is happening because of excessive inflammation. So we're only trying to sort of take care of the excessive damage. Of course, there's no specific way to measure it. We have data from pathology as there is secondary inflammation, and so that's how we know there is likely damage because of that. Yeah.
You want to say a word about long-term steroid being sometimes causing more weakness?
Yeah, so one other caveat is that if you are on steroids for a long period of time, apart from helping with the inflammation, that itself can cause damage to your muscle fibers. Again, leave you with some patterns of weakness, for example, weakness around your hip muscles, that again might be difficult to reverse because there is damage to the muscle membrane and the muscle itself, different from autoimmune diseases or genetic diseases causing muscle injury. So when you see your physicians, especially if you're on steroids, we try to be cautious of how long somebody is on steroids, what are the potential side effects of it, and could they be developing this condition called steroid-induced myopathy. These are all the reasons I worry about using steroids forever. We try to look for more specific therapies. But steroids do work. I mean, for myasthenia, probably no drug works as well as steroid. And I usually tell my patients, if steroids did not have side effect, myasthenia as a field would not exist because there is no need for any specialist. Anybody can take a steroid and the disease would be controlled. So.
And just to add that from I know there's parents who have kids with Duchenne in the audience, and you worry about the long-term use of steroids also. And there's multiple longitudinal international studies that show the greater benefits of steroids despite all of the side effects in both muscle strength function and lung function and cardiac function long-term.
This is a perfect time to ask Dr. Bennett this question. I have been surprised how long Ionis has been in existence. The question is, how long does it take from drug development to FDA approval?
Yeah. So it's there is no fixed timeline. It really depends on the project and how things work. And so I can use SMA as an example just to give you some experience or a reference. So we started working with our collaborator at Cold Spring Harbor on spinal muscular atrophy in roughly 2004. In 2011, we dosed our first patient. I remember it was right before Christmas in 2011. The first patient that was ever dosed with an antisense drug for spinal muscular atrophy was dosed. We finished the pivotal clinical trials in 2016, so roughly 5 years later, or 4.5 years later, we had the data that we needed to support going forward into to the FDA to get approval. That was took us about 3 months, or our partners Biogen, 3 months to pull all the data together, write up the study reports, and submit them to the agencies. And then 3 months later, the FDA approved the drug. So it's really, that was probably one of the fastest approvals in FDA history. But they saw the benefit and the need for the patients, that they worked extremely hard to get the approval. Typically, it would take the FDA 6 to 9 months, sometimes over a year, to approve a drug once you submit all the data. And so I would say on average, once you start a clinical program, you're probably looking somewhere between 5 and 10 years in that range, depending on the disease and how big of a trial you have to do to be able to get the data you need to submit. And then generally it takes the FDA 6 months to about a year to approve it. One of the other things I will mention is that if it's a rare disease, that can be sped up. If it's a common disease like Alzheimer's, they're going to be more cautious, and it's going to take more patience, and it's going to take longer. And so it does really depend on the disease and also how well the drug works.
That 3-month approval must have been a record, I think. Is it?
I think there's one other drug that was a cancer drug that was equally as within the same time period. But it really is a record.
I think I just speak for how dramatic the results are. I mean, those of us who have seen since the treatment result, there was absolutely no doubt. Questions for Tina. How often would you recommend exercising? This is a loaded question. Because that I suspect is going to be need to be individualized. And this question asker also talk about yoga, and for this person, that yoga has changed the life of this particular person. So, two questions here: exercises and yoga.
I agree. I definitely can't give you, I feel like I'm sitting here just saying, I don't know. I can't give you an answer to the exercise because that is very specific. I highly advocate for exercise to be implemented for everyone, regardless of your functional level. There's always an adaptation for you to be able to exercise. The ACSM statement states that you should have 30 minutes of exercise 3 to 5 times, of cardio exercise 3 to 5 times a week. And I always advocate for you to work with your therapist to make sure that plan is individualized to your diagnosis. Because there's certain neuromuscular diseases that you can't strain the muscle fibers too much because it doesn't regenerate well, and so you want to make sure that your program fits within the paradigm of safety. And then yoga. I am a big fan of yoga, in particular because it's a combination of stretching and strengthening. For the most part, if you're not talking about power yoga and more the sun salutations and things like that, there's a couple things it addresses that's very core. One is trunk stabilization. A lot of times when we talk about stretching and we're talking about limbs, but the movement is it's important to have strong trunk stabilization. If any of you've ever sat in a room with me, that's going to be the first thing I tell you. Because if you have a strong core to work off of, then your limbs, your arms and your legs can move better. And so yoga focuses on that, which is crucial. And if you're in a wheelchair or if you're walking, that is something that is relevant to everyone. And then the isometric strengthening piece is a safe way to strengthen muscles, and they do that quite a bit through yoga. So I'm a big fan.
Do you want to talk about, occasionally we get this question in clinic, should I just buy some dumbbells and start doing weight training?
No. The basic answer is no. So any kind of strengthening program, there's really, with different neuromuscular diseases, like I said earlier, is based on the pathology. So there is not a flat answer all over the board about strengthening. There are some muscle dystrophies, such as myotonic muscular dystrophy or CMT, in which resistive or strength training is not detrimental to muscle health, while Duchenne, for example, is one that you don't want to put that kind of stress and strain on the muscle because it does cause fiber damage that is not reversible.
And analogous question. Once a contracture has happened, can it be reversed?
There's no evidence that once a contracture has occurred, contracture meaning it's fixed and there's no flexibility in the movement, from stretching or any kind of conservative management has not been shown to change that range of motion, and the only way to make a change in a fixed contracture has been surgery. And that being said, surgery for contractures should be a multidisciplinary team approach because you don't want to go to the orthopedic surgeon. He cuts the tendon, makes your joint look perfect, but then because of that elongation of the tendon itself, now your muscle fibers have to work harder to contract, and therefore you may lose that function. So you want to talk to a larger team beyond just the orthopedist when you're considering something like a surgical tendon release.
This is a generic questions about stem cell therapy in neuromuscular disease. That wasn't covered in this morning's session. Maybe we'll open to our experts here. Any thoughts? I know Ionis is not involved in stem cell, but you probably have thought about that.
So I think it's experimental therapy, and so I think there are a lot of questions that we all have as investigators and scientists about what stem cell therapy is going to be doing, and I think it's still very early days. I don't have any personal experience with it, and so I defer to my colleagues at the table or other people in the audience that may have a little bit more direct experience, but I just view it as very early.
I mean, I know there is an ALS trial for stem cell. I don't know whether we are a site, but I know CPMC.
No, we're not. We're going to cover the stem cell in ALS more in the breakout session later in the afternoon. Suffice it to say that it is early. It's in the proof of concept stage. And also, for the most part, when we're especially dealing with neurons like SMA and ALS, stem cell therapy is not just a replacement part. I think that's a common misconception, is that, well, my neurons are dying, and we add some stem cells and replace what's dead. That's not how stem cell is likely to be beneficial if it turns out to be beneficial. So stem cells really just, hopefully, if all works, improve the environment of either the spinal cord or the brain to make the neurons, the nerve cells, the motor neurons live longer. So whether that can be pulled off with injections and whatnot, that remains to be seen.
So kind of a derivative question is gene therapy, and so there are a couple of questions on gene therapy, and again, it looks, in particular for SMA in the infants, it looks very promising, and as a potential therapy. The question that I got, the specific question is, can it be combined with antisense therapy? And the belief is theoretically, yes, it can be. There are going to be some financial challenges, obviously, but there's no reason that you couldn't treat somebody with a gene therapy and also an antisense type drug for perhaps getting a better overall response to the therapies.
Okay. Well, I'll keep Frank on the mic for one more minute. This is a question about the site of the intrathecal injections. Is there data about cervical and cisternal injection? This is, as you know, spinal tap usually is done in the lower back. This question is whether there is any reasons to inject it higher up.
Yeah, so there are some SMA patients who are getting cervical, which is the base of the skull injection. Generally, it's a more technically demanding procedure, and I think it's mostly done by interventional radiology. So it's a different, because of the challenges. But I do know that there are some patients who are getting cervical.
That's often done out of necessity, because especially with development of scoliosis and curvature of the spine, sometimes it's just physically not possible to do a spinal tap. How about what you're thinking about how well does the medicine diffuse up and down the neural axis?
Yeah, so what we've so in experimental animal models like non-human primates and dogs, we see that once you introduce it into the cerebral spinal fluid, that's the fluid that surrounds the brain, you get good distribution throughout the neuroaxis. We've done some studies where it's not something that we're recommending doing, but where you've done into the ventricle, which is into the brain, kind of a fluid compartment within the brain. We've seen very similar distribution as what you would see with lumbar puncture into the lower back. So I suspect the distribution is going to be similar, but it really hasn't been studied, and it's something that we need to do as a community to study this better.
Okay. I think time is up. There are still a lot of questions that are not, I didn't have a chance to put out. Some are probably better directed more individually around lunchtime to our speakers. I trust that they will be available to all of you. So, Jennifer, you want to tell us what to do next?
So, thanks to all of our speakers. If we can give a round of applause. And thanks to Dr. So, who stepped up and helped out. So we're going to be serving lunch, and it's going to be outside. We'll open up the doors, and you can sit wherever you would like. You can sit outside. There's lovely fountains out there. The weather is beautiful. If you don't know what you ordered, check with one of the staffers. She's got a list, and so there's turkey, pasta. Sorry, I screwed up I said there was chicken. It's actually turkey. And there's pasta, and there's a veggie sandwich, and you should have an initial on your name tag. And if you don't, just check in with the Stanford staffer, and they will tell you what you ordered. And if you are coming back here for the motor neuron ALS SMA breakout session, we will be back here at 1 o'clock, and all the other breakout sessions are down the hall. So have a nice lunch.